Hello everybody, welcome to Tuesday, January 19th, because remember yesterday was MLK Day, no school, so now we're starting this week on a Tuesday. Great for you to join us here today. I'm sad that you're not here in person, but hopefully you'll be with us in person pretty soon. What we're going to do today, first off, bell ringer as always is going to be up there. We are looking at mutations, and we're going to look at, there are 6 kind of mutations that we're going to look at. 3 of them we're going to look at today, 3 of them we're going to look at tomorrow. There's a little bit of an assignment that goes along with each one. So, without further ado, let's take a look at what we have going on here with these things. Here is the folder for today. You can see the bell ringers up there at the top. You've got a copy of the mutations, just the blank notes of it. So if you have a printer available, you can always print them out at home. Otherwise, you can also just copy them over and make it your own document. You can go up to the files for it and copy the document, and then you can have it in Word and type in there if you want to do it that way. Or you can take them on a piece of paper if you want to also do it that way. There's a couple of different options for that. And then down below, you'll see the slides for the mutations. Those are the notes that we're going to take a look at today. So let's pull those up. I'm getting it loaded here. Mutations. There we go. Full screen it up. What we've got going on for mutations here. First off, our definition of mutations. We already kind of talked about it before in the past. It is a mistake or change in the DNA. Let me shrink myself down a little bit. There we go, shrunken down. So a mistake or some kind of change in the DNA. It's not always a mistake. Sometimes it's just a change, and some changes can be good. Like if you were a cow and had laser eyes, that'd be pretty good, I would think. Laser-eyed cow. Obviously, mutations are not going to be like that, but there are different causes for a mutation that can occur. Our causes for mutations can be one of the following things. They can be spontaneous. Oh, it kind of looks like. Oh, I didn't plan this, but it kind of looks like I'm the Hulk. But first off, going back to what we had here, mutations can be spontaneous, which means they just happen. Sometimes we can't control it. We're doing everything right. Mutations can just happen. And as we looked at, there's a lot of DNA. The chances of a part getting messed up is pretty high, but the outcome of it, those are the things that actually control whether or not we actually see it or not for the mutations. Another thing that could be causing a mutation is what we call a mutagen, which is anything that can cause a change from our environment. And these are things like chemicals, radiation, radioactive substances, X-rays can cause it. Ultraviolet radiation, which is sunlight. You kind of get the idea that there's a lot of stuff dealing with radiation and or chemicals a part of it. Those are 2 big things from our environment that can cause this. What it can lead to then are different things like if the person is pregnant and comes into contact with this, it can lead to things like birth defects. It could lead to things like in older people, things like cancers can develop because of a mutation. And other varieties of different diseases and disorders can come into play. What it does not lead to are superpowers. So the 3 effects of mutations that we have, they can be negative, which means that they cause some kind of problem. We don't want negative ones. There can be positive ones, which are very rare, which make something just a little bit better in some way or another. Or what a majority of them actually are silent, which means we don't actually see them. They're neutral. There's nothing going wrong with that. And that's because, as one of the videos talked about last week that we watched with the Amoeba sisters, a lot of mutations might happen in that non-coding region, the stuff that is shut off. Because your active DNA is going to actually be pretty low in each one of your cells. Because if you're, for example, a skin cell, you don't care about the sections of DNA that are going to be making your heart, or the sections of DNA that are going to be making up your stomach, because stomach cells and heart cells deal with that. So you're only activating the DNA inside of your cell that is going to be needed for whatever cell that you are. If you're a skin cell, it's only going to activate the parts of DNA that are relevant to a DNA or for a skin cell. If you're a hair cell, it's only going to activate the parts that are going to be necessary for a hair cell to know. It's not going to care about the other sections. So sometimes, or a lot of the times, it'll happen in that area where there isn't a whole lot of DNA to begin with. That's actually active because you're only focusing on such a small portion of your entire DNA. I think they said somewhere between 3 to 7% is your active DNA. So a huge majority of it is going to be your non-active stuff, the stuff that would be relevant in other cells. And because not one cell needs to know every single little bit of information from the DNA, some mutations just go unnoticed because it's not going to affect us. So here's a little bit of a preview about mutations and some of the mutations that will take place. You'll find this video in the Schoology if it does not work in the slideshow. And then let's look at our 3 mutations that we're going to talk about today. Our first mutation that we're going to look at is a deletion mutation, which basically means that stuff gets cut out. We remove it. So there's different ways that we can look at this. It could be a removal of a single base, like in our sequence over here. Let me get my little pointer out. There's my little red pointer. It could be the removal of a single base on here. Here's this T in the original DNA. We just pop that out. Why this is a problem is that it moves everything over one spot. So all of our proteins are going to be mutated after that point. It's not going to make the same thing. Or it could be a much larger chunk that gets removed. So here's the whole entire chunk of DNA. We could take out an entire chunk. So we're missing this blue part all of a sudden. So it could be just one nitrogen base that gets removed, or it can be an entire section of DNA that just gets lifted. And then you don't have that DNA anymore. It's just lost at that point. Some examples of what would be a deletion. One of them is color blindness. So with a deletion, you just do not have certain proteins that can register certain colors for you. So that's what's considered color blind. You just don't have those proteins anymore onto it. And there's a variety of different types of color blindness. I've got an example of a colorblind test here. If you're not colorblind, you should be able to see a number appear in the circles. In this case, if you haven't figured it out yet, that's the number 8 that you should see. I'll kind of draw with the outline. If you are colorblind, all of these would look like the same color because in your eyeball you do not have the protein to specify what is the green versus what is the red. And there's other ones. And there's just other varieties of them to do different color sensitivities to things. So this one on the left side here should say 27. This one on the right side says 42. Got the 4 there and then the 2 happening there. Another example of a deletion would be cystic fibrosis, which basically means extra mucus gets produced in there. It is a deletion mutation that causes this. It leads to a bunch of problems developing to you, typically around your lungs because there gets mucus in the passageways and things get clogged, so your airflow isn't going to be as good. And then I tried to find a wide variety of mutations, some common, some maybe not so common. But there's a rare blood mutation that is caused by a deletion that causes your skin tone to become blue. Kind of like the Blue Man Group there. So you got some kind of blue mutations going on there. Second one is that insertion. It's the opposite of deletion. Instead of deleting something, you get something added into it. So here we added in another section of it. Additions to a chromosome from another chromosome, or you just add in some more bases to it, but you're adding something to it. Some examples of addition or insertion mutations is albinism, or being an albino. The lack of skin pigment is actually because you have more stuff added into it. And specifically, we have tracked it down using our science knowledge and the fact that we're making proteins. Specifically, the TYR amino acid that you make, that's one of our amino acids that's on our chart, that is the one that is specifically getting disrupted in there, that is causing you not to produce the type of pigment that you should be producing. And this happens in a variety of different types of animals there. So you can see reptiles and at least mammals there for it. Basically anything that has pigment can suffer from being an albino. Is that on the wilder side of things? Insertion mutation. These are warts that were formed from an insertion deletion that caused this man to get epidermodysplasia verruciformis, which basically had warts that developed bark-like qualities because it just toughened his skin to be almost about bark-like in quality. And they just kept on growing and growing and growing. Eventually this man had him removed. And when he had him removed from after here, he had about 11 pounds of these warts removed from his hands. But they can grow and develop anywhere on your body. Usually they start pretty early on, but they just continuously grow and grow and grow and grow. Obviously, there's different ways to affect them. You could have gotten them removed earlier. He had them growing out of control there at that point, though, to where he had these growths on his hand. And then another one that's kind of a positive spin on it is with bacteria, where they can digest nylon, which we've actually used that as humans to help eliminate wastes in water. Because nylon, like this type of fabric, it's not biodegradable really. It stays around a lot because it has elements of plastic to it. This bacteria developed this mutation where they can actually eat nylon and dissolve nylon. So we have used it in some of our waters to treat that. And our final mutation that we're going to look at today is substitution. That's where we just exchange one base for another or one section for another. So we lift one portion out, add something else in. It could be one letter, like we swap an A for a G there, or it could be an entire section gets lifted out. So here's an entire section moving to it. Some examples of substitutions. This would be like sickle cell anemia. It causes your blood cells not to be circular, but rather like more of a moon shape, which means for that sickle shape, that gets clogged and stuck in your veins easier, which is bad. You're not as good as getting oxygen flow throughout your body. Another example is with TaySachs disease, which basically is a neurological disease that affects your brain and spinal cord. The substitution goes on in there to substitute different elements in your brain and spinal cord development. And as you can read there along the bottom, usually it's diagnosed very early on because it goes along with a baby's ability to be able to turn over or to sit up. They lack some of that control that goes on there with it. So there's our kind of 3 mutations to wrap it up there. Now I'm going a little bit long, but there's an activity that goes along with today. Let me pull that up real quickly and go over it with you. Where would my stuff be here? Let me just find it. Just pull it up again because I don't know where. I probably exited it out earlier here. So let's look at science. We'll go all the way to January. Down here to mutations day 1. Your homework will have to deal with mutations, and you're going to answer a couple of questions along with applying some of those mutations as well. So you got some drop-down options there. They do always go in the same order, so the first couple are always going to be your standard message or your standard notes questions there. The special ones like this one here, you are going to use this codon chart, which is a little bit different because we're going to see how this method or how these things get affected by showing you how it would work if we were to build out a word. So here you'll find these sequences and write down the word down below here. So we got our start, and then you're going to write our word. You don't actually have to write out start, but you'll write down our word. So you'll find G-U-G, first base G, second base U, third base G. We got a capital H. So we'll put a capital H in there. We'll go to your third one, U-U-C, U-U-C. It's an A, and you'll finish out the word. You're then going to do the same thing down below. You'll notice that a lot of them are the same, but you'll notice that there is a mutation that might happen to one of them. That is the thing where you specifically would need to lock on. A note from the original to the mutated. Make sure you put it in there correctly going in. You don't have to put the start part in there, but when it has a period or an exclamation point, make sure you add your period or your exclamation points in there. And then there's questions about the things that came earlier. What was the things that happened in question number 5? You'll have another one where you'll look for the mutation. There's basically a couple of sets of these going on in there to code for a specific type of mutation that we just looked at today. So it has a little bit of application processes to it. And like with all of these, you can take them multiple times. So if you get it wrong, you can retake it, relook at it again, see what you did right versus see what you did wrong. And always retake it to improve that score. But this is your assignment then for Tuesday. And I already have looked at, taken up a lot of your time here, so I'll wrap things up here. If you have any questions, email me. Otherwise, your assignment is on there to look through. I hope you're doing well, staying safe, and I will see you hopefully in class tomorrow. If not, I'll see you right back here for Wednesday's video. See ya.